tardes. Hoy tenemos el gusto de tener al doctor Steven Herbert con nosotros dentro de la serie de conferencias que presenta la Cátedra de la Fundación Miguel. He pedido al doctor Herbert, que fue su estudiante durante algún tiempo, con el que ha mantenido una observación extraordinariamente a lo largo de los años. Que nos haga una presentación del doctor Herbert.
Gracias. Pues tenemos el gusto de tener hoy entre nosotros al doctor Steven Herbert, gracias a que fue invitado por parte de la Fundación Miguel Alemán para dar la cátedra Miguel Alemán y pues aprovechar para algunos otros seminarios importantes. Steve es actualmente el jefe del Departamento de Fisiología Celular y Molecular en la Universidad de Yale. Yo conocí a Steve hace 17, 18 años cuando llegué a su laboratorio para hacer mi postdoc y trabajamos juntos durante 3 años en el Hospital Brigham and Women's de la Universidad de Harvard en Boston. Y desde entonces pues hemos mantenido una relación muy estrecha de colaboración científica y pues ahora de amistad que ha sido pues razonablemente productiva. Y Steve pues es un investigador que es médico, se entrenó como médico en la Universidad de Florida y después hizo su especialidad en medicina interna y en nefrología en la Universidad de Alabama a finales de los 70s, y ahí conoció a uno de sus principales profesores, el doctor Tom Andrioli, con quien se fue a la Universidad de Houston por algunos años, y junto con él hizo las descripciones iniciales de los mecanismos de transporte de sal en algunas de las partes del riñón, particularmente en el asa ascendente de Henle. Ahí describieron por primera vez ellos los mecanismos de transporte, particularmente de la membrana apical, en donde está el transportador de sodio, potasio, cloro y el canal de potasio, que es probablemente su molécula favorita. Después en Boston se dedicó algunos años a terminar de describir estos mecanismos de transporte, y para el momento en que yo llegué pues vino el cambio total en su laboratorio. A lo que nos dedicamos fue a clonar todos estos genes, lo cual fue posible y ha traído pues una cantidad importante de avance en los conocimientos a nivel de fisiología y fisiopatología renales y en otros tejidos. De las 3 áreas donde él ha sido particularmente exitoso, una de ellas es los transportadores electroneutros, otra en el área del sensor de calcio, que fue de lo que habló ayer en la Fundación y hoy en la mañana en el Instituto de Nutrición, y una de las áreas, la tercera área, que es probablemente su favorita, es esta área de los canales de potasio. Entonces lo que nos va a hablar hoy es de estos canales de potasio. Después de haber trabajado muchos años en la Universidad de Harvard y llegar a ser full professor, se movió inicialmente en la Universidad de Vanderbilt y después ahora en la Universidad de Yale. Y de lo que va a hablar ahora es de los canales de potasio. Steve.
Thank you for the invitation and being here. I'm happy to be here, and I want to tell you about one of my favorite proteins, in large part because I cloned it while at Harvard, but we were interested in body potassium homeostasis. How do we maintain our potassium balance? And it was clear and well understood that the kidney was vital to maintaining potassium homeostasis. And we knew that there were potassium secretory processes which would move potassium from the cell, having come in from sodium potassium ATPase, move it into the cell, and then allow for its secretion into the urine, and therefore it could be excreted in urine, and you could help maintain body potassium. And we were interested in what that potassium channel was, what was the molecular nature of that potassium channel. And at the time we started this project, around 1990, there was only really one class of potassium channels that had been identified, the voltage-gated potassium channels. The amino acid sequence was known. It was a 6-membrane spanning protein. And so when we began this project to identify these channels, and it was known that the potassium channel in the kidney was a weak inward rectifier, but it nonetheless was an inward rectifier. It passed current better into the cell than out of the cell. And when we identified and cloned this from the rat kidney, it was a new structural model for a potassium channel at that time in about 1993. And it contained only two membrane spanning domains. This is the tetramer of the channel, so it forms mainly a homotetramer, but there are channels that are formed from two different genes and two different subunits. It forms, there are four subunits, and these four subunits come together and form the pore, the potassium pore, which is down the center plane. And the ROMK potassium channel that we call for rat outer medullary potassium channel, because it was cloned from this particular region of the mammalian kidney, the rat kidney. And we knew that this region of the kidney expressed a large amount of this particular channel on the apical membrane of the thick ascending limb in the kidney. And it was called KIR for potassium inward rectifier 1 because it was the first gene to be identified of the inward rectifiers. And right at the same time, Lily Jan in California had been cloning another inward rectifier, KIR2. And it was a strong inward rectifier, and I'll tell you what we mean by strong and weak rectifiers in a moment. And actually, her paper in Nature came out about 2 or 3 months after our paper did. In fact, she was a reviewer on our paper, and she asked if she could include some things in her paper because our paper just hadn't quite come out. It was a week or two from publication, but that was the start of the field. And then since that time, very, very rapidly, KR3, which are the G protein beta gamma regulated potassium channels that are present in heart, the ATP gated or regulated potassium channels present in a wide variety of tissues, CNS, vascular, heart, and even in the kidney. And this was in a very, very hot field for a while because identifying the ATP-sensitive potassium channel in pancreatic beta cells was kind of a holy grail for many people working in diabetes, because this was a key channel that regulated a process for insulin secretion. And so it was really quite important. And the channel turned out to be really quite complex, and I'm not going to talk about this channel, I won't describe it, but it involves an interaction with an ABC protein, a so-called sulfonylurea receptor, because it is the binding protein for sulfonylurea compounds, which are very active in treating some forms of diabetes. But it's a complex channel. So this is the family members. If for potassium channels, it's a moderate size. It's not a large family of ion channels. And again, they're inward rectifiers, and they're either strong or weak. So the strong inward rectifiers have little to no outward current. All their current is inward. And they're very important in stabilizing membrane potentials towards EK, and are found in a wide variety of cell types, including neurons. And then the weak inward rectifiers have a significant outward current, and that's obviously very important. If we're talking about a potassium channel, it's important for potassium secretion from the cell into the urine. It better pass an outward current. If it's not passing an outward current, it's not secreting anything. So it is a weak inward rectifier, and we understand a lot about what is causing this inward rectification. A large part of the inward rectification is because there are cations, particularly calcium, magnesium, and polyamines that can come into the inner part of the pore of this channel and block it in a voltage-dependent way. So as the current is passing in this direction, these molecules come in and actually plug up the pore. And that's what creates either the strong inward rectification or the weak inward rectification. So this is one of the monomers of the tetramer. And this is from work that was done by Declan Doyle. Declan Doyle did his postgraduate work in Rod McKinnon's lab in New York. And you're probably aware that Rod McKinnon, a few years back, won the Nobel Prize in chemistry for his structural identification of a number of ion channels that allowed us to begin to look at and peek into the molecular mechanism of ion permeation and gating. So this is the bacterial potassium channel, and you can model ROMK or any of these inner rectifiers by putting it on the backbone of this structure for the bacterial channel, and the fit is quite good. And all of the inward rectifiers basically look quite similar to this. Here's the amino terminus. It comes around to an alpha helical region. When we first cloned ROMK, we identified this alpha helical region as an amphipathic region. And we had a bit of a time and a bit of a debate in deciding whether or not this was a membrane spanning domain or not a membrane spanning domain, because it was very clear that we were going to have either 2 or 3 membrane spans, and it was clear this was going to be a new structural model for an ion channel. This was relatively short, and I think we decided that it probably was interacting with the membrane, the inner leaflet of the membrane, and wasn't membrane spanning. There were so many other reasons for convincing ourselves that this was in the inner leaflet of the membrane and wasn't membrane spanning, and our guess at the time was right, based on current structural models. Then you come into the first membrane spanning domain, go through there's a pore helix and then an area that is in the selectivity filter, and then you come back down to M2, and then you form this complex array of beta strands that form a bit of a basket area that's shown here. And one of the very interesting aspects of these channels is that there's an interaction site between the amino terminus and the carboxy terminus, but not in the same subunit. So the carboxy terminus in this region here interacts with the amino terminus in this region of the adjacent subunit. So there's a subunit interaction. And a few years ago, we identified, as well as a number of other workers, particularly Fran Ashcroft's group in UK, that it is this interaction region between subunits of the amino terminus of one subunit interacting with the carboxy terminus of the other subunit, which was the binding site for ATP in the ATP-gated potassium channels, KR61 and 62, that are found in heart and pancreas beta cell. So this was a very interesting region between the subunits that was seemingly playing a role in gating. Now the model for gating is still not entirely clear, hotly debated, but one of the aspects that appears to be important, and there's accumulating data that this is the case for ROMK, is that there's in the inner part of the vestibule of the pore, there is a meeting, this so-called inverted TP, and you see it here, where the alpha helices cross, and there are amino acid residues in this region that appear to absolutely plug the pore. And if you mutate these to smaller residues, you can actually eliminate gating. The channel is open all the time. Now, there are also glycine residues in M2, and what we think is happening is that this slide helix has a degree of freedom axially within the inner leaflet, and when it moves outward, it allows for kinking of M2, and the kinking of M2 is important for rotating the first membrane span out of this crossing bundle, and therefore opening up the pore. There's a fair bit of data in supporting this. The selectivity filter, there's a lot known from the bacterial channels about the selectivity filter. It appears that there are 4 binding sites for potassium in the selectivity filter. And at any one point, 2 of these sites are occupied. That's the best data that we have in the field. So it's either 1,3 or 2,4 binding. So those sites on average are bound. So within the selectivity filter, at any one time, you see 2 potassium ions, okay, binding at site 1 and 3 or binding at 2 and 4. Now if we go to the kidney, this is the cortex of the kidney, the outer part of the cortex of the kidney. The medulla, outer medulla, is here, and then this is the papillus, and where the inner medulla would be. And the outer medulla is in this region, and that's where we cloned the potassium channel. This is in situ hybridization. You can see there's a lot of message for this. There's also a message within the cortex, and these are particularly in tubules of the mammalian nephron that contain cells that are actively involved in potassium secretion. Although it's not quite clear here, this is apical staining of this potassium channel, and it decorates the apical membranes of the cells, and it's in a position, therefore, to be important in secretion. Well, if we look in more detail of where this channel is expressed, it's expressed in 2 regions. One of them is the thick ascending limb, and this ascending thick limb is very important in allowing us to make concentrated urine or dilute urine. It's the site in which there is actually separation between salt water and the mammalian nephron. So what happens here is that this apical membrane is virtually water impermeable, and this transport mechanism actively reabsorbs sodium chloride out of the tubular urine, therefore reducing the tonicity of this fluid and creating free water generation. It's also important for countercurrent multiplication, the mechanism that's necessary for our ability to concentrate the urine by increasing the osmolality of the interstitium of the outer medulla and inner medulla. So the transport mechanism is through this triple ion transporter that Dr. Gamba, when he was with me, cloned and identified, and has been working on ever since. We called it BSC for bumetanide sensitive cotransporter, but in general, most people refer to it as NKCC2, or the sodium chloride potassium cotransporter. And its electroneutral involves 1 sodium, 2 chlorides, and 1 potassium. So the sodium enters, moves to the blood interstitial side through the sodium potassium ATPase. Chloride moves through the base lateral chloride channel. And the potassium recycles across this apical potassium channel, which I'll show you later is identified as this ROMK protein. This is a very active, many hundreds of millimoles of potassium are recycled across this channel every day. And so this, having this channel here is very, very critical. It's critical for the operation of this cotransporter for salt absorption. And the reason for that is the load of sodium that comes to this region after being filtered at the glomerulus and some fraction being reabsorbed in the proximal tubule. So as sodium enters this region for reabsorption, the quantity of sodium that's presented there every day is many fold higher than the quantity of potassium that's presented. And this cotransporter has a 1-to-1 stoichiometry for sodium and potassium. So if you're going to absorb any sodium, you need to have an equivalent amount of potassium. And the delivered load is not equivalent. So this K recycling is absolutely mandatory for absorbing the load of sodium that is necessary for maintaining sodium balance. Now, in the more distal parts of the nephron is where there is active and regulated potassium secretion. And this has, on the apical side, this would be facing the tubular fluid, has 2 major transporters, and I'll show you there's a third one there that's important in a minute. So there's a sodium channel, and this is the epithelial sodium channel. And this was cloned by Bernard Rossier, Cecilia Canessa, and their colleagues, about the same time that we were floating ROMK, which is this K secretory channel. And sodium absorption is really an important component that's required for full and active K secretion. And the reason for this is that sodium entry, as you can imagine, depolarizes the cell. And the depolarization of the cell is a critical driving force for subsequent potassium secretion through its ion channel. So there's a link, an electrical link. I don't know why this keeps stopping, but there's an electrical link between sodium absorption and potassium secretion. So this is the region where there's active K secretion. There's K secretion here, but it's involved in this recycling process, which is a critical factor for sodium absorption in the thick limb. So if you look at a human glomerulus of the kidneys, they filter about 180 liters of fluid per day. Okay? So you can think of 180 liters of normal saline a day. And 20% of the filtered load of sodium is reabsorbed within this region. So the absorption of sodium is huge, and this recycling of potassium is huge in this region. So we knew that we cloned ROMK. We knew that it was expressed in these very important regions for potassium secretion and recycling. And over the now about 15 years since ROMK was cloned, many laboratories, including our own, have been working on this and looking at the regulation of this potassium channel. And the regulation is very complex. And this is a small protein, about 297 amino acids, and virtually every amino acid is doing something in the amino and carboxy terminus and involved in regulation. And all of the classical processes that you can imagine involved in regulation are involved in regulation in this protein. So there are regulators of exocytosis and regulators of endocytosis, and this endocytic recycling mechanism is very important in setting the level of potassium, or the number of potassium channels in the luminal or apical membrane. This recycling process is predominantly a clathrin-dependent process, although not exclusively. Okay? But the predominant mechanism is clathrin-dependent. There is also a very important part of exocytosis, and I apologize, this arrow should go in this direction. The exocytosis, the movement from subapical vesicles of potassium channels into the membrane. And there's a very important process that is regulated by kinases. And the way that these kinases work, either serum and glucocorticoid regulated kinase or protein kinase A, is that they phosphorylate the amino terminus of ROMK. And this phosphorylation of ROMK is critical for blocking an ER retention signal on the carboxy terminus of the protein and allow forward movement out of ER to the apical membrane. So we published this in PNAS just about 2 years ago as a phosphorylation switch for forward trafficking of a protein. There actually have been several other examples of that, but this was really a very interesting example. So the carboxy terminus has an ER retention signal, and so you make the protein, but it doesn't get out of the ER unless you phosphorylate this site. If you phosphorylate this site, it allows for forward trafficking. And we hypothesized that this was an important part of the process of recognizing that the channel was folded properly. And I don't have time today to go into that, but if anybody's interested, I'll be happy to answer any questions.
Well, excuse me, if
Yes.
you have a metabolic syndrome, a lot of corticoids in blood, this traffic is increased, and then that helps to hypertension or something like that?
Yes, so we have, there are mechanisms. For example, if you increase aldosterone and you increase the level of SGK, you'll increase the number of potassium channels on the apical membrane. And we believe that an important contributing part of having more apical membrane channels is that you have a greater rate of exocytosis from ER through Golgi, post-Golgi into the apical membrane. So that's an important part of the process. It's a slower process, which is aldosterone increasing K secretion is a slower process. It doesn't occur in minutes, it occurs in hours. But it's an important process of the overall regulation of the channel. There are many factors that regulate the open-closed state of the gating of the channel. One of the things that we call gating God in this channel is the intracellular pH. This is extraordinarily sensitive to intracellular pH, and much has been written about this. Many papers have been published about this. And one of the predominant or primary mechanisms by which the channel gates is by a pH. Normally in these cells, the pH is not changing all over the place. Like most cells, the pH is guarded very closely. It wants to keep a set pH, doesn't want to modify or change it. So then if you have a gating mechanism that's sensitive to pH, how can changing pH gate? Because the pH really doesn't change in the cell. And what happens is that the EC50, or the set point of the pH sensitivity, which normally is around 7.1 to 7.2, is modified by all these other factors that affect gating. So PIP2 level in the membrane, kinases, even ATP can affect the set point of pH. So basically what you do is you don't move the pH, but you move the pH curve. And that's how you modify a large part of the gating of this channel. But the common pathway through which many of these kinases and factors work is through modification of the pH sensor and sensing. Many people have looked at this. We've looked at it. It doesn't appear to be a single one or two amino acids that regulate pH. There isn't a discrete pH sensor. We've identified, and many other people identified, probably a set of 10 amino acids that are all over the place in the protein that are involved in pH sensing. So we don't think of this as a pH sensor that is a discrete region that interacts specifically with proton, but it involves multiple sites within the protein, all of which are being modified by the proton concentration and are affecting the gating behavior of the channel. So it's a diffuse sensor, more like modifying the isoelectric point of the protein.
Is exclusive for KIR1 channels?
No.
So
So not all of the channels are highly sensitive to pH, so there is a subset, but there are other channels within the KIR family that are sensitive to pH. So it's not just ROMK. Yes?
Yes. And are these amino acids spread out toward the amino or the C-terminal?
Both. Both. So this was something that disturbed us for a while. I mean, there was a number of early papers that suggested that there was a triad, 3 amino acids, that came together and formed a sensor. And then when when Declan Doyle at Oxford got the X-ray crystal structure of the bacterial channel that we could then model on ROMK or any of the other KIRs, it was very clear that these amino acids, which do affect pH sensing, were scattered. They were nowhere near each other. They weren't connecting by salt bridges. They were completely independent. And so that led to the concept of not a discrete pH sensor, but it's actually the titrable sites scattered across the carboxy and amino terminus, and it's the summation of that leads to a structural modification, slight probably, that affects gating behavior.
Yes, I have another question about the tyrosine phosphorylation of this channel.
Yeah.
They are so involved in the exocytosis, right?
Yeah, so that's I've listed up here. So protein tyrosine kinase and protein tyrosine phosphatase are very important regulators of endocytosis. And in fact, this is a predominant part of the mechanism that is modified by dietary potassium intake. Okay? And it is the connection between dietary potassium intake and the regulation of the number of channels on the surface of the apical membrane. And the process works that in a high potassium diet, that protein tyrosine kinase activity is very low in these cells. The channel isn't phosphorylated. The channel isn't phosphorylated and therefore doesn't recycle, doesn't go back into the cell. So there are more channels on the membrane because endocytosis is reduced. In a K-deficient diet, there's a lot of protein tyrosine kinase activity. Phosphorylates the channel, more endocytosis, fewer channels on the apical membrane.
But at which point in the trafficking pathway is tyrosine participating in phosphorylation?
Yeah, we don't know. I mean, we don't know the binding partners, okay? But all we know is it requires phosphorylation of a particular carboxy terminal residue, tyrosine residue on ROMK, on the channel itself. And this somehow signals for endocytosis to occur. So you have to have an intact endocytosis mechanism. For example, if you use a dominant negative clathrin, you can stop the process. Okay?
Are the channels in the lysosomes or in the end?
Oh, the channels are at the membrane when this is occurring. The channels are at the membrane and active when this is occurring. And then they're endocytosed. So there's still a lot of work that's going on in this area. This is, as you might imagine, this is a potassium channel that's regulating K secretion. Therefore, our ability to get rid of potassium or hold on to potassium in the body. And you can imagine there might be many, many, many different regulators. And in fact, there are many kinases, many factors that are involving gating and regulation. Not only gating, but the average level of potassium channels on the membrane. A large part of the regulation is not so much in gating. And the reason for that is that the open probability of this channel is 0.9. So this channel is open virtually all the time. That's great if you're secreting a lot of potassium. Okay? So it's either on or off. It's a switch. So most of the channels are on, and they're active when they're in the membrane. You can switch them off, and there are some mechanisms I don't have the time to talk about that can switch them off in the membrane. Okay? It's a very intriguing mechanism. But mostly the channel is active when it's in the membrane. Most of the regulation is by trafficking. Okay? 90% of the physiological regulation is here. And again, WNK kinases, protein tyrosine kinase phosphatases, SGK, PK, all involved in a complex way to regulate this trafficking process. Well, I want to talk a little bit about, and the rest of this, to really talk about what this potassium channel is doing and what its potential relationship to blood pressure. So this is just a very brief clinical digression, but there are a number of genes that have been recognized by Rick Lifton and his group and others, including our group, that are involved in the regulation of blood pressure. And they regulate blood pressure by regulating salt reabsorption or salt excretion by the kidney. And this provided really quite important evidence for the role of the kidney in blood pressure regulation. Now that doesn't dismiss that there aren't important vascular effects. But how much salt we retain in the body, how much salt we excrete by the kidneys or retain by the kidneys is a critical factor in blood pressure regulation and the development of hypertension or hypotension, reduction blood pressure. So a number of genes have been identified that can lead to high blood pressure, low blood pressure, and these all involve very important salt transporters, basically sodium and chloride transporters along the nephron. So these are the basic mechanisms that allow for regulated salt absorption out of the filtered urine back into the blood. Okay? They include this sodium K-chloride transporter that I talked about. There's another co-transporter here, the sodium chloride co-transporter that Gerardo Gamba, when he was in my lab, identified. And then there's this ENaC sodium channel that, as I said, was cloned by Cecilia Canessa and Bernard Rancière in Lausanne, Switzerland. So these are the three very important mechanisms for salt absorption. But there are other players within the thick ascending limb, which I'll show you in the next slide, that are important for regulating salt absorption by this thick ascending limb. When any of these 5 genes are mutated, it leads to a renal phenotype of salt wasting. So you can't retain salt by the kidney very well. It's like having a loop diuretic, like a furosemide diuretic. These individuals are hypotensive. Okay. So what happens, and this syndrome is called Barter's syndrome. Sometimes it's called hyperprostaglandin syndrome because one of the phenotypic characteristics are a high prostaglandin in the blood, and prostaglandin E2 in the blood and in the urine. But these individuals waste sodium, so remember about 20% of the filtered load is reabsorbed here, and if you don't do that, then you're excreting a large amount of sodium in the urine that normally you should retain to maintain blood pressure. So there's salt wasting, there's a reduction in effective circulating volume, and this leads to a reduction in blood pressure, an activation of the renin-angiotensin system, increased aldosterone. And with activation of the renin-angiotensin system, there's activation of the prostaglandin production, particularly prostaglandin E2. So what are the genes that are involved in here? So there are now 5 genes that have been identified that can lead to this renal sodium wasting phenotype. And you can imagine that mutations in this cotransporter would do it, and in fact, that is the case. You could imagine that there's a chloride channel that's very important for chloride movement across the cell, and mutation in these channels would result in the phenotype, and that's true. There's also a single transmembrane spanning domain protein called Barton, and Barton is a critical protein for trafficking these chloride channels into the membrane and having them available for chloride movement. So if you eliminate Barton by a loss of function mutation, these chloride channels are made, but they can't make it to the basolateral membrane, and therefore they're not functional. So this gene is important in trafficking. This is the chloride channel. Mutation of either of these will cause this salt wasting because you can't absorb chloride. I talked a lot in the previous 2 talks about the calcium sensing receptor, and I don't want to spend time with that here, but there are activating mutations that basically can downregulate salt transport in a very complex way and can cause Barter's. But what I want to talk about today is ROMK. So loss of function mutations in ROMK cause Barter's syndrome. So you can stop sodium chloride absorption by reducing or eliminating this potassium channel. And how is that done? Well, I told you that there is a very important K recycling, and this is important for providing substrate for cotransporter in equal proportion to the available sodium. But there's also another important aspect of this transport process. If you look at the stoichiometry here, you see 1 sodium crosses into the cell, but there's 2 chlorides. And most of this potassium recycles. So where is the other cation coming from? And the other cation actually is going through between the cells, through the paracellular pathway. And this is an extraordinarily electrically leaky epithelium. And what's important for that process is a current loop that involves chloride secretion basolaterally. So that's a current going from basal to cell. And then K going from cell to lumen, that's this recycling K, is current flowing in this way. So there's current flowing from the basal side of the cell to the apical side of the cell. And then the circuit is completed through this electrically leaky epithelium by carrying this positive current through the paracellular pathway. And what makes up that current is the other sodium that's absorbed. So when you eliminate ROMK, you not only eliminate the recycling, but you eliminate this current flow pathway. And so equivalently, you stop flow of chloride basolaterally. It's just an electrical circuit, and basically I make an infinite resistor here, and you stop the flow of current. Okay? So this is a very potent effector of sodium chloride absorption. Okay? So modulation of the activity state by all the factors that I talked about earlier, of this potassium channel plays a very important role in regulating sodium chloride absorption, okay, through this process of requiring recycling in this current flow pathway. So potassium, this potassium channel is involved in recycling, as I indicated, is involved in secretion. So ROMK, we and others have proposed it as a potential target for a diuretic, particularly a potassium-sparing diuretic. And why is that? Because if I block the activity of this potassium channel, I would have a drug that would be equivalent to blocking this co-transporter like furosemide. But I'd also block K secretion, and therefore I might have a situation where I could lose a lot of sodium without having an increase in potassium secretion. Now, why would you have an increase in potassium secretion if I blocked a lot of sodium here? Well, the sodium that's blocked here from being reabsorbed goes distally through these nephron segments where the potassium channel is. And if I increase sodium reabsorption, I increase the depolarization of the cell, move potassium away from EK, and I will have more potassium secretion. So the more sodium I present here, the more potassium secretion I have. So most diuretics are waste potassium as well as sodium chloride via this mechanism. So if I had something that blocked sodium reabsorption in the thick ascending limb, which would be a very potent diuretic, but at the same time blocked K secretion in the distal nephron, that I might have a potassium-sparing diuretic, which could have some functional and physiological advantages for treating a whole variety of disease studies. So that was what really kind of led us, one of the things that led us into looking at this in much more detail. So we would anticipate that we might have individuals who have a ROMK barters that might be hyperkalemic. They would have a high body potassium because they don't have this potassium channel to secrete. And in fact, when these infants are born, they do get hyperkalemic, but only transiently, only actually for a few days. And then they actually develop hypokalemia. Their body potassium stores go down. So that was very curious. So it suggested that there must be some other mechanism besides this K secretory channel for moving potassium out of the body into the urine and having potassium loss. So that was another reason that we wanted to look at, is there some other process that's causing K secretion? What is that process? And can we learn anything from that? And the way that classically you would begin to do that is to make a knockout mouse. Yes?
Yes, do ROMK channels distribute in other tissues?
They are present in the brain. No one that I know of has looked at that yet. They are present, and we looked at this very recently, in the stomach, and they are involved in, there are one of the channels, there are several channels, it's one of the channels involved in apoC K recycling from the gastric HK pump. So if you knock out ROMK, you actually reduce acid secretion in the stomach. And we've demonstrated that, and we're about to submit that for publication. So it does play a role, but the predominant role that it plays is in the kidney in K secretion. There are a couple of other places where it appears to be playing some role. So we went ahead and made this model. And the model wasn't made by this guy, Ming Liu, but Ming did a lot of the studies. And he's an important person. And he does some things that I'll tell you about now that are close to magic, technically. And he's been doing it now for over a decade. And he's invaluable, and you can't recruit him. So we were doing this knockout at the same time Gary Schull was doing the ROMK knockout. So we combined forces and got this ROMK knockout. And the null mutants had a lot of sodium wasting. They had reduced sodium absorption in the thick limb, which you would expect, and a number of other metabolic parameters that I won't go into details with. but are something that you would expect given this mutation. One of the troubling things that came about from this is that these kidneys were very hydronephrotic. What we mean by that is that there appeared to be loss of renal mass with dilation in areas where there's no cells, there's no nephrons. And in fact, the glomerular filtration rate of these kidneys is low, and these animals died of renal failure. And how bad was this? Bad. So the initial survival was about 2 or 3% post-weaning. And so that's not a great model. That's a lot of money and a lot of work to get a few animals. So are there ways to fix this? So one of the ways is to say, look, you've got a mouse, and this mouse survives weaning. So you could cross it with other mice, but we chose another way, and that was to do crossings of survivors. And we've now done that over the last 3.5 to 4 years. So there are many generations out, and they're not absolutely gene pure, but they're getting closer and closer. So what did that do? Well, that increased survival. You can say, gee, this isn't great survival, but if you want to look at these animals, this is when you got 30% survival. Well, it's even increased now to about 50%.
What is the reason?
And that's, we don't know. So we don't know the genes, and we haven't had time to. It'd be very, very interesting to look at that. What are the survival genes here? We just haven't had a chance to go through and look at that, but we hope to sometime over the next couple of years to begin to look at that in a little bit more detail, because I think that may tell us some interesting things. But we now had enough animals that we could work with. Now the problem is that the matings are heterozygotes. Okay, so you have to breed a lot of animals to get your knockouts, and not all of the homozygotes survive, at best only 50%. So a lot of breeding. The advantage is that you have all the controls. So you have gene-matched, wild-type, gene-matched heterozygotes and with the homozygous knockout. So there's actually an advantage, even though it's a lot of breeding work. So what does Ming-Lu do? So Ming-Lu can take kidney from a mouse, dissect out individual tubules, so individual segments of the nephron. He can then take those and, with cell tack, attach them to a cover slip, take a glass knife and split them open so he has access to the luminal face of these cells, and do either cell-attached or detached patching to look at single-channel activity, kinetics. And it's a very tedious and not so easy process, but when you've been doing it for a decade, he can do it on even small mice, sick mice. He's been a tremendous asset in doing this, not only in this model, but a number of other models. So we looked at that. So we looked at this both in the thick ascending limb and the collecting duct, which is this distal nephron segment involved in K secretion. And in the wild type, every time you make a gigacele patch, you don't see a channel. And that's not atypical. So about 2/3 of the time, you see a channel. So that's the baseline. And that's true whether you do a wild type rat, a wild type mouse. Not every time you patch do you see a channel. And these are gigacele patches, so they're not leaky patches. So this is the norm. About 2/3 of the time you'll see channels. If you take the heterozygotes, lo and behold, about half that. If you take the knockouts in either of these segments, no activity over many patches. Okay. So these are these 30 picosiemen channel conductance channels that we and many other people had identified in these nephron segments that were involved in K recycling and K secretion. And so this provided, I think, the definitive evidence that indeed ROMK was the molecular counterpart of this channel that plays this important physiological role. This is a small table, a lot of work. You can look at the numbers of patches here. So in doing this process that I told you about, and but a necessary process to say that yes, indeed, ROMK is this channel. Now, what about the mouse? What is the phenotype of the mouse? Well, they excrete more urine. They're diuretic. They're natriuretic. They excrete more sodium. They have an inability to maximally concentrate the urine, which is what you would expect. They excrete a lot of prostaglandin, which is what you would expect. And they have an increased loss of calcium. And I don't have time to go into this, but this is the same phenotype that we see in patients. So these patients with Barter's don't waste magnesium, but they waste calcium. And so this is going to be an interesting model in order to look at what's going on in the distal part of the nephron that's giving this particular phenotype. Yeah.
What happens if you block prostaglandin synthesis?
Yeah, in humans, if you do that, the children get markedly better. There's less sodium wasting and less potassium wasting. And we're beginning to look at that now in these mice. It's not so easy to do in mice. One of the best COX inhibitors, cyclooxygenase inhibitors, it causes ulceration in the stomach. So you have to, it's not an easy thing to do. It sounds very easy. Just give them a COX inhibitor. So we're looking at COX-2 inhibitors. They're a little bit better. They're still not so easy to administer, and dosing is a problem, but we're looking at that to lower this. There's an interesting phenotype here in that the prostaglandin excretion is 2 to 3-fold higher in females than males. And it's something that we're very interested in, and we're looking at that. There's not clear evidence that there's a worse phenotype in females than in males in the human disease. But it's a very difficult thing to study because most of these, there aren't big, large families that you can study. It's sporadic. And so it's just not easy to do that kind of analysis. So one of the things that we wanted to say is, does ROMK knock out lower blood pressure? And it does lower blood pressure in these animals. What about potassium? Well, their potassium is is a little bit on the low side, but not greatly so. Below 3.5 is clearly hypokalemic, but there is a significant difference between wild type and ROMK knockouts. So they're within the normal range, but on the low side of the normal range. So they have the kind of classical phenotype that we would see in patients with this type of Bartter syndrome. And so we wanted to now look at, all right, what's going on in this model? You know, are there any aspects of survival that we can identify? And so there is a type of survival mechanism that you might think of, and that is they are losing 20% of their filtered load of sodium because they're not reabsorbing it in the thick ascending limb, but maybe they're increasing the mechanisms of salt resorption more distally along the nephron. So the next most distal nephron segment is the distal convoluted tubule. It begins right after the thick ascending limb. So if you take the wild type animals and you look for the sodium chloride cotransporter, you see it. And these are these distal convoluted tubule segments. And all the images I'm showing you here are at the same magnification. Okay? So you can see these tubules. Here's the lumen and here's the outside. This is apical staining. My God, look at these things. These are huge distal convoluted tubules, and I'll show you some EMs of those in a minute. If we look at the, I'm sorry, I made a mistake. So this is just a higher magnification of the wild type. So here is the large guy right here. So you should compare these two, and you should compare these two. So this is a ROMK knockout, and so these tubules look bigger on average than these. And in fact, if you look at the fractional cortical volume occupied by the distal convoluted tubule in the knockout, it's several-fold higher. So the volume occupied by these enlarged tubules is much greater than in the wild type. If you look more distally, the connecting segment and the collecting duct are downstream of this distal convoluted tubule. They're not expanded in volume. So it appears to be a specific enlargement and expansion of the distal convoluted tubules. And so if you compare this at higher magnification, what you're seeing is the apical border. The tubule is around here, the cells around here.
Why is it so right?
Well, because this is a cell, they're just bulging out into the lumen. So this is a cell, one cell would be here, and this is the nucleus. I'll show you, you can see this in an EM in the next slide. Okay? So these are long tubules. So, sorry. If you look at the EM, so again, these are EMs. This is the micron bar. This is the same magnification. This is a normal distal convoluted tubule. This is the distal convoluted tubule in this knockout. They're huge. And the cells look normal, but there are more cells, okay? And the cells are a little bit larger. So this is hyperplasia and hypertrophy, both. And we're very interested in identifying what are the factors that are involved in this hypertrophy. Okay. Now, I don't have time to go into this this afternoon, but again, this is a very, very fruitful area of saying what are the factors that regulate the growth and postnatal development of this very important region of the kidney that's very important in regulating sodium chloride absorption. And by regulating sodium chloride absorption, regulating extracellular fluid volume and blood pressure. And so it had been known for a while that there were factors like a high salt diet that would expand this region. And people said, oh, maybe it's sodium movement into the cell, chloride movement. There was some data that suggested that angiotensin II through AT1 receptors was basically acting as a growth factor in the cells. But very, very difficult to tell. Here we have gene-matched animals, both homozygous, heterozygous, and wild type, that we can begin to look at what are the specific factors that might be involved in this expansion. The other extreme of this, and I'll get to your question, just one statement and then. So the other extreme of this is there are knockout animals where the co-transporter, the apical sodium chloride co-transporter is knocked out. When you do that, this cellular mass virtually disappears. So there's some suggestion that maybe its sodium entry into the cell somehow is being recognized and acting as a growth factor or growth inhibitor. So more sodium chloride seems to be coupled with more growth. Less sodium chloride and less growth. To the extreme that if you knock out the co-transporter, cell mass disappears. Okay? If you expand the amount of sodium chloride that's being absorbed here, maybe it's acting as a growth factor. So we're very interested in looking at how that's signaling in the cell, how it's recognized, and how that translates into growth of these cells. Yes?
How is the DCT in those mouses? Have to check that die after weaning?
We've only looked at that in a couple of papers a decade or so ago with some colleagues in Europe and in Florida, University of Florida. And we looked at this by acutely reducing sodium chloride entry into the cell by using a diuretic, a thiazide diuretic. And this was done in rats. And so what happens if you give rats a diuretic, and you can do this by either injection or by an osmotic mini pump, over a period of a few days, and then you look at the animals over this period of a few days, what happens is that there is cellular apoptosis. Okay? And so there's actual cellular loss by apoptosis. And again, another example that's suggesting that sodium chloride entry is somehow signaling as a signal for growth of these cells. Now, this is not universal. I mean, if you give a loop diuretic, which shuts down NKCC2 in the thick ascending limb, and you can do this over weeks, okay? The thick ascending limb is still there. All the cells look normal. There's not really a great deal of change that's occurring in the cell. So it's not a universal phenomenon that if you have a salt-transporting epithelium or cell, and you shut down salt transport, that all of a sudden the cells disappear. Some cells could care less. Thick ascending limb doesn't care at all. Shut me down, I'll look the same. Okay? But if you do that to a distal convoluted tubule, it cares a lot, it seems to care a lot. So again, this is, I think, a model that is going to be fruitful in looking at what are the factors that modulate the cellular mass in a region that's critically important for salt absorption and regulation of salt homeostasis and blood pressure. So is there really salt absorption going on? So you see a big tubule, you see a lot of co-transfer. It doesn't mean necessarily that there's a lot of salt reabsorption, but in fact there is. So if you take these animals and you look at the wild type and the knockout under control conditions, well, there's the natriuresis that you see in the knockout. And if you add furosemide, you don't see anything different. In fact, the furosemide animals come up to what the knockout animals are, consistent with the knockout ROMK is actually like furosemide, just you're diminishing salt transport in the thick limb. That's where furosemide blocks NKCC2. But what if you block with thiazides? Well, here's the change from here to here in the wild type. Look at this huge sodium wasting that occurs if you block salt transport in the distal convoluted tubule, this NCC sodium chloride co-transporter portion. So this region is actively reabsorbing sodium chloride that's not being reabsorbed in the thick ascending rim. So one of the survival factors is the ability to get a robust upregulation of the distal convoluted tubule, and where you can make up for some, not all, but make up for a lot of the salt wasting that's occurring in the thick ascending rim. You just make it up here. The wild type animal can't do that very well, but the knockout animal, possibly through the selection process, has upregulated this region, regardless of whether or not this is a natural or a selected phenotypic characteristic. It provides an interesting model to look at in the context of many things like what are the growth factors, what's the role of salt transport in growth and remodeling of this nephron segment. Now, what about further downstream? So if you go more downstream here, this is where ENaC, this sodium channel, is being expressed, is in this region. What's happening there? Is that upregulated at all? So what we can say is that, yes, there is upregulation. This is Western blot, semi-quantitative Western blot staining, wild type knockout, big upregulation of the alpha subunit, little possible upregulation of the beta, but a big upregulation of the gamma. So the sodium channel is made up of 3 subunits, an alpha, a beta, and a gamma. These are separate genes, and they make up, probably with an stoichiometry of 5, 2 alphas, 1 beta, 1 gamma, although that's still a bit controversial. If you look at ENaC mRNA, particularly the alpha subunit, there doesn't appear to be an increase in messenger RNA. So this appears to be either translation that's upregulated or stability of the protein is being upregulated, but it's not a gene effect, it's a post-gene effect. And we haven't figured out exactly where the upregulation of protein expression is, whether it's stability of the protein or whether it's a higher rate of translation. But we're looking at that now. But we wanted to look at whether or not, there's a lot more transport protein, so maybe this region of the nephron is also absorbing more sodium and chloride and helping out the knockout animal. Well, that's not the case. So here's a set of animals. You can see this robust response to an NCC transport inhibitor. But when you add benzamyl, which creates a natriuresis in the wild type that's quite significant, there's no difference in the knockout. So despite this big upregulation, there's more sodium channels that are being made, more protein that's being made. That's not translated into a physiological effect on salt transport. So what's the possible explanation for that? There could be many, but one certain possibility is the fact, remember that I told you that there's an electrical coupling between sodium absorption and potassium secretion. So what would happen if I eliminated this potassium channel? Could there be sodium absorption? The answer is yes, there could be. Sodium can come in. But what would happen in this case? The sodium entry would tremendously depolarize the cell, reducing the driving force for sodium entry. Normally, this is counterbalanced by K secretion, which repolarizes the cell. So now you've lost the bulk of this repolarization, and now you have sodium entry. And we think that this is playing an important role in reducing the potential sodium absorption and not increasing it above normal. In fact, there's a hint that it might be a little bit less. It's not statistically significant, but it doesn't make any difference. It's not upregulated.
Well, so
Yes.
there are also other explanations, right?
Yes.
You can also have other channels involved in that.
Yes. So I'm going to tell you about another channel that's here just in the last couple of minutes. So remember, there's still a problem here. If ROMK functions to promote K secretion, then why aren't these animals hyperkalemic if they got to have a K secretory mechanism, okay, somewhere in these cells? So we then searched for that because that was important. So we had a hint that there must be a K secretory mechanism in this nephron segment by looking at the excretion of potassium. So if you give hydrochlorothiazide in a wild-type animal, so this is sending more sodium distally, so there's more sodium to be reabsorbed. This electrical coupling means more K secretion. So in a wild-type animal, you reduce absorption here, downstream you see huge increase in K secretion. Okay? Do you see that? And this is change from control. Okay? The big increase. Do you see this in a knockout? Yes, you do. So there's got to be a K secretory mechanism. Now, where is that occurring? Is it in the same nephron segments where ENaC is expressed? And the answer is it looks like there is. That if you add benzamyl, which blocks these sodium channels in a wild type, there's less K secretion. Okay? Because now there isn't a driving force for K secretion because you're not depolarizing the cell. Do you see that in the knockout? Yes, you do. You see a reduction. Okay? Again, there's got to be another K secretory mechanism in these cells. And so there was a channel which had been suggested, and that was the maxi-K channel or the BK channel. Okay? And indeed, it is expressed in these nephron segments. Difficult to patch, and we have an explanation for that now, but difficult to patch. And so the question was there BK channels that were active in actual K secretion in this region of the nephron, particularly in the knockout animal? Now, the BK channels can be activated by increases in intracellular calcium, among other factors, depolarization as well. And one of the factors that's been postulated is flow sensing, a flow sensor, which is this central cilium. And for aficionados who know polycystic kidney disease, the polycystic kidney disease genes are actually expressed at the tip of this central cilium. And the central cilium in these cells has been suggested to be a flow sensor. It's still an area where there's a bit of controversy, but at least there is certainly a fair bit of data which is consistent with this being a flow sensor. And it's been shown in cultured cells that if you move this central cilium, that you can result in an increase in intracellular calcium, which could activate these BK channels. So this is a flow sensor, but the channels could be activated by a wide variety of other kinds of mechanisms as well. So we did want to look at whether or not this channel was involved in K secretion. And iberiotoxin is a really great drug for blocking these channels. And iberiotoxin has, okay. So how do you go about doing this? Well, the only way that you can really do this is by getting together a dream team. And we did that. And this included people who were in their 70s, in their 50s, and one young guy. And Matt Bailey was the young guy. It involved Gerhard Malnick from Brazil, Gerhard Giebisch from Yale, and Tong Wong from our Yale group as well. And this involved in vivo micropuncture in a process that involved blocking the filtration of fluid in this nephron, having a perfusion pipette where you could put fluid of a known composition into this distal part of the nephron, taking that column of fluid between oil blocks, so you have an isolated column of fluid. Moving it to a position where there is a double-barreled microelectrode that's measuring potassium activity, and doing this in an alive anesthetized mouse, transgenic mouse. So you can imagine that this is a tour de force and required really the gods of the world able to do this, the experts that have been doing this for decades. So we were really fortunate and able to do this. And what you do is you perfuse in a solution that has half millimolar potassium, and you look at the rise of this potassium over a period of time. And kinetically, you can model this and get a potassium secretory flux. And I won't describe this in any more detail. It's a bit complex. It takes 10, 15 minutes just to describe the process. But it's a very reproducible process. Very difficult to do, but possible to do. Iberiotoxin doesn't affect ROMK, so we needed to know that. ROMK just is not blocked by iberiotoxin. So any iberiotoxin-sensitive K secretion is not ROMK in the wild-type animals. It has to be these BK channels. So we looked at that using this, and this is on a normal potassium diet. And so this is the rate in nanomoles per centimeter squared per second, and the centimeter squares per surface area. And this is the secretion rate, okay? 0.65 nanomoles per centimeter squared per second, okay? And if you look in the knockout mouse, it's about half, okay? It's not zero, okay? There's no ROMK channels here. We've patched that, okay? But there's still K secretion. What about iberiotoxin? Well, in the wild-type animals, you can block about half the K secretion with iberiotoxin. What about in the knockout? It's all gone with iberiotoxin. So these knockout animals have expressed these BK channels, which become K secretors. They're not fabulous K secretors. They have very low open probabilities even when they're active. But they have large conductances, and they allow for a significant amount of potassium to be secreted. And so this is the principal mechanism for the K loss that occurs in this ROMK knockout mouse. And we suspect is a contributing factor to the K loss in all forms of barters. Of course, in the non-ROMK knockout barters, there's still ROMK there, and ROMK can act as an important component of secretion.
Sorry, and how do they respond with the high diet, potassium diet?
I'll show you that next. Okay. So if you take the animals and you put them on a high K diet, okay, what happens? Okay, so this is 1% diet, this is 10% diet. This is the ROMK part because this is the sensitive part, this is the BK part, this is the ROMK part, okay? BK goes way up, okay? ROMK goes up a bit, but the largest fraction of increase is BK. So in a very high, this is a huge K diet, but it's typical of what we give rodents in modeling. And so the upregulation that occurs, and this is in the wild-type animals, and we've done this in C57 BL6 mice as well. So this is not just a funny ROMK wild-type species effect, that in the C57 black 6 mice, you see the same thing. There we see absolutely no increase in ROMK, and all of the increase is BK.
Does it mean more times more BK?
Yeah, so good question. Can I go on to the next slide? Okay. So it means more BK channels in the membrane, a bit, but primarily it means an alteration in the gating of these channels. And with Van Nuy Wong, who's in Medical College of New York, who we've been collaborating with for a number of years, he's looked now in some detail at these BK channels in wild-type animals. So this is wild-type animals. And so one of the things that had been known actually for a while was that if you take a rabbit or you take a rat and you put them on a 1% diet or a 10% diet, and you then use antibodies to look at what the expression level is of BK channel, what you find is that the expression of BK goes up in a high K giant. So there's more protein available for making channels. And but what's important is the gating, and how do we know that? So if you take a normal K diet animal and you patch the principal cell, you rarely find BK channels. Rarely find BK channel activity, patch it. Okay? The protein's there. Okay? You don't see the channels open. In fact, it's difficult even to open them if you load the cell with calcium. You get a little bit of change, but it's not huge, loading with calcium. And we found that the reason for this is that the channels are inhibited by MAP kinases, particularly ERK and P38. And that these MAP kinases, we don't know if they're phosphorylating the channel specifically. All we know is there are two MAP kinases are involved. And why do we look at MAP kinases? Because we knew that there was the modulation of MAP kinases that was occurring with diet. We, with Louis Wong, had looked at that, as well as protein tyrosine kinase. And in fact, it's the upregulation of MAP kinases that activates protein tyrosine kinase to be an effector of ROMK. So if you look on the normal K diet, here's phospho P38, here's phospho ERK, 1, 2. What if you're on a high K diet? Okay, about the same amount of protein, maybe a little bit less, but the phospho is gone. What about phospho ERK? Almost gone. Okay. And again, this is another replicate pair. This has been done a number of times. So it's a very reproducible phenomenon. So if you're on a normal K diet or a K deficient diet, your steady state activity in these cells of MAP kinases is elevated. And this inhibits BK. On a high K diet, and by mechanism we don't know yet, that this reduces the phosphorylation. Okay? There's a little down regulation, but it's not really much. I don't even think it's of any significant consequence in total P38. But there's a big reduction in phospho, P38 or ERK, that occurs in this condition. So you don't have activated P38, you don't have activated ERK 1, 2, and having these kinases appear to be very important in whether or not you have significant activity of BK.
So then how do you explain in the knockout, the ROMK knockout, that these channels are functional?
Yeah. So we don't know the answer to that yet, and we're looking at it. So we're just now in the process of looking at whether or not there's an alteration in P38 and in ERK expression or in phosphorylation in the ROMK knockout animals. We're also looking more quantitatively at the number of BK channels in these animals. This is a process. It takes a year or so to do these kinds of studies. There's a lot to do here, but it says that there is a partner for ROMK, for K secretion. It's a particular channel that is turned on during a high K diet. ROMK does participate, but it's not the only player in K secretion on a high K diet. It's under a normal K diet, it's a predominant player. Okay? But this comes in as a fail-safe mechanism to allow for K secretion if we eat a high K diet. Keeps us from being hypokalemic. Okay? Now, I'm not going to show you data because my postdoc would go crazy if I showed data because he hasn't published it yet. But we do know that ROMK is a critical player and an important player in K secretion in a high K diet. So one of the ways that we can ask this is to knock out ROMK, but a specific isoform. So I didn't go into detail, but there are 3 isoforms of ROMK, called ROMK1, ROMK at the beginning of the indeterminate. And ROMK, this distal aldosterone expression in these nephron segments. There's also a bit of ROMK2 here, so they do have some potassium channels. Okay? So if you look at these animals under a normal K diet, they're have a slightly elevated but not abnormal potassium limb. So if you knock out ROMK1, you don't get Barter's, but you knock out the ROMK1 expression in these nephron segments. There's also a bit of ROMK2 here, so they do have some potassium channels. Okay? So if you look at these animals under a normal K diet, they have a slightly elevated but not abnormal potassium. But if you put them on a high K diet, they get profoundly hyperkalemic compared to the wild type. So we know that both channels are important. But I'm not diminishing BK. BK is very important on a high K diet, but also ROMK is important on a high K diet. And what BK does is it contributes to the overall K conductance property of the apical membrane, allowing you the potential of secreting much more potassium. And in the situation where ROMK is gone, in the mouse model, it can account for the K secretion that we're seeing that's still occurring in these animals, okay, despite the absence of ROMK. And it is sufficient, okay, although lower than normal that I showed you, but is sufficient to not allow these animals to become hyperkalemic. And in fact, probably is contributing in the ROMK Barter's humans to the hypokalemia, the actual hypokalemia that's seen in these individuals, although it's mild. Okay? It's much more mild than, say, in an NKCC2 knockout, which delivers the same load of sodium distillate, but now there's ROMK and BK to secrete potassium. They get profoundly hypokalemic because they have all the potassium channel array to secrete masses of potassium. Okay? So this type of Barter's is the milder form of Barter's as far as K wasting is concerned. It's because part of their major machinery is gone. This part is an interesting aspect of it, and I think has potential relevance to BK expressed in many, many other cell types. That there's a very critical and important regulation by a set of MAP kinases. And Wenhui Wang is vigorously looking at what's the downstream of this and even what's the upstream of these MAP kinases. We know a part of the upstream is the tyrosine kinase. But we don't know that that's the major or all the upstream of that in this situation. So this is the end, and I wanted to tell you a little bit about what this channel is doing and how we've begun to look at in using a whole variety of techniques over a period now of about 12 years or so, in trying to understand what this potassium channel is doing as one model for the inward rectifiers. It's certainly a model where you can't use all the findings in all the family members of inward rectifiers, but it's telling us some fundamental properties. And we know that if you get rid of ROMK, you reduce salt transport and some K reabsorption that might occur in here. But you can continue to have K secretion by these BK channels, and the BK channels are upregulated both in expression and gating by the high K diet. Now, one of the interesting things, and I'll just use this as a finish, and one of the things that's interesting is how does the animal perceive an increase in or a reduction in dietary potassium intake. This is actually an extraordinarily interesting and fundamental biological question that's not answered yet. There are a number of groups that's looking at it. What we can tell you is that if you feed an animal a high K diet, they don't get hyperkalemic normally. So it's not an elevation in plasma potassium that's keying the kidney into hugely changing all of these systems to allow for enhanced secretion and therefore excretion of potassium in the urine. People have begun to study where that is. My bias, based on the limited information that's known in the literature, is it's not the kidney. It is either in the GI tract, in the portal system, or in the liver. And people are vigorously looking at that. And there's some evidence to suggest portal system, and there's some evidence to suggest maybe the GI tract. And so remember, dietary potassium, before it's excreted, has to be absorbed. It has to go through the portal system. So the question is, are there sensors? Are there something in there that's recognizing the intake of potassium, beginning to change K secretion as soon as you start to absorb that K load? So remember, these changes in the kidney that are required for enhancing K secretion don't occur instantaneously. They can take hours. So if you take a K load, as soon as you take in that K load, you might think that it would be some advantage to already signal to the kidney, get ready for this load because you're going to have a lot of K to get rid of. And so physiologically it does make sense, but and people have actually looked at this possibility for 40 years or more. There's data going back to the 60s in this area. And again, it's an interesting model or question with regard to overall homeostasis, because homeostasis begins at whether we absorb or don't absorb something from the GI tract. And having the GI tract signal to other organ systems could be important. And we already know that there's a huge number of factors that are produced by the GI tract. And there are, I'm sure, factors that aren't even known yet that are produced by the GI tract, because it is the primary site of, hey, something is happening here. I've got this factor or that nutrient or this ion, and I'm absorbing it and telling the body about what's going on at the entry site of all of these factors. So it's going to be, I think, an interesting decade as we begin to really look at, okay, how does the kidney know that there's a change in dietary intake of potassium? So these are the players, Minglu, Qidong, Kerr, we call them, Cheng Lang, Li Yitang, and Qing Shang Yan. So you can say, you have a nice Chinese group here. And I have to say that Yale University in the U.S. was the first university in the U.S. to graduate a Chinese citizen with a PhD. And when was this? In 1870. Okay? 1870. Okay? The first Chinese citizen to get a PhD in the United States, Yale University, 1870. So we've had a long-standing relationship, over a century of interaction with China, and now we have an interaction with them that has actually been codified and in contract. So we are starting this year getting about 15 of the top Chinese graduates that are handpicked out of 3 of the top universities in China to come to Yale to do their graduate studies. So this has been a long process, but we expect to have some more great Chinese minds to come to the U.S. and help us out unravel the mysteries of many of these things. Gerhard Giebisch is a longtime collaborator. Tong Wang is a woman who's a wizard in doing in vitro micropuncture, in vitro microperfusion. Rick Lifton's lab has been involved in many studies related to the Wnt kinases, as well as Gerhard's lab. And others, particularly Gerhard Malnick and Matt Bailey, were very, very important in doing the in vivo micropuncture, which was the first study that established that the BK channel could actually be an effective K secretory channel in the mammalian nephron. So thank you very much for your attention.